Hello everyone, I'm Charlotte from MDPI and I will be your host for today. Thank you for joining our latest MDPI webinar on cancers entitled Epigenetic and Metabolic Alterations in Tumor Microenvironment. This is the second part of this webinar series based on the special issue from our journal of the same name. If you are interested in submitting your paper to the special issue that is linked to this webinar, please check the webinar's webpage. I would like to remind everyone that this webinar is recorded. The recording will be made available as soon as possible and will be posted on the webinar's webpage. We are also live streaming this webinar on MDPI's YouTube channel, so hello to those of you watching us on there. Furthermore, certificates of attendance are available upon request. We can only prepare certificates for those of you that are registered and watching on Zoom, not on YouTube. Today's webinar has two chairs, Professor Ciro Isidoro and Professor Dhanasekaran. Professor Isidoro is from the University del Piemonte Orientale. His laboratory has a consolidated experience in the biogenesis and function of lysosomal proteases, and in alterations of the autophagy lysosomal proteolysis in cancer. Emphasis is given to the study of protein-protein interactions and to the subcellular localization of the protein of interest in order to understand its altered function. Professor Dhanasekaran is from the University of Oklahoma Health Science Center. His current research is focused on defining the role of coding and non-coding RNAs in tumor microenvironment and tumor progression, along with a focus on developing novel cancer therapeutics. Thank you to both of you for accepting to chair this webinar. 4 presentations were originally planned for today, but given the problems that were encountered during the webinar yesterday, there will be now 5 presentations. Given by Professor Dhanasekaran, Professor Ciro Isidoro, Dr. Hervouet, Dr. Umber, Cheema, and Dr. Rainer Glauben. Each presentation will be followed by a Q&A moderated by the chairs. If you have any questions, please feel free to post them in the Q&A box at the bottom of your screen, and make sure to put the name of the person you're asking the question so we don't get confused. Now it's time that I hand the microphone over to the chairs. So enjoy the webinar, everyone.
Thank you very much, Charlotte, for the introduction. I would like just to introduce you with this. And as Charlotte mentioned, this is linked to a special issue of cancers, which is managed by Donna Xu. I wish to thank her. And the guest editors are myself with Danny. We will have just a change in the schedule. I will give the lecture at the end if we have some time left, just a short presentation. So I would like now to introduce my co-chair, Danny. And Danny, after his studies in India, he moved to United States, and he is presently the professor of cell biology at Oklahoma City University, and he is deputy director for basic research of the Stephenson Cancer Center. He is also our visiting professor here in Italy. He is also visiting professor at Seoul National University in Korea and at Tokyo Medical and Dental School University in Tokyo. And today, he's going to give us a lecture on long non-coding RNAs and cancer epigenome. So please, Danny, it's now for you. Thank you.
Thank you, everyone. Especially, I want to thank the MDPI cancers, for providing this opportunity for this very seminar on this popular topic. And also, I think I want to thank Ciro for taking this huge burden of organizing this enterprise. I think without him, probably it would have been extremely difficult to host this webinar in this critical time. Um, Without much more on that, what I would do is I'll start my talk. And unfortunately, I again apologize for not able to present yesterday because of the technical problems. But what I would do today is I'll kind of, I have, shorten my talk so that, we'll have, time for Chiro's presentation at the end as well. So as Chiro was mentioning, my topic today is, link RNAs and cancer epigenome. And our lab has been working on the non-coding RNAs for, quite some time now. So we have recently interested in how long non-coding RNAs are regulating the cancer epigenome. Okay, what is epigenome? The epigenome is actually defined by the huge array of chemical tags, a finite array of chemical tags rather, that provides directive to the genome whether particular gene is turned on or off. And in fact, the good definition is, there is a recent review in which, the DNA is organized in the chromatin form, multi-epigenomes, and that, specifically it requires the, DNA cytosine methylation so that, codon modifications can define whether the particular gene is on and off. This is regulated by both in the, post-translational modification in the histones as well as in the DNA. And if you look at that, there are, several players in that, and colloquially or casually they are being called as writers, readers, and erasers of the epigenome. You have a family of protein that involved in putting the methylating tag. In many cases, that can turn off the gene. That is, methyltransferases in specific metallohistones. And there are erasers, that is, lysine demethylases in the histone. Likewise, you have methyltransferases that can put the tag in the DNA, and erasers, especially the new family of TET proteins that can erase these signals. Then you have other writers in the involved in histone methylation, in the histone tails of the genome. In addition, you have several specific domain that acts as the readers for these writers and erasers. The new player in this, the readers, writers, and erasers are the polycomb group of proteins. They actually act as the regulators. I would rather consider them as the kind of facilitator for specifically modulating or facilitating the writers, erasers to kind of go to the specific sites so that the genes can be appropriately modified. Recently, what it has been identified is that most of these PCG proteins, you have two complexes. One is polycomb repressive complex 1 and polycomb repressive complex 2, and they are involved in they are major players in this the epigenetic silencing in cancers. However, what is known is although that they are involved in the repression and the repression mediated activation of some oncogenes, all those things happen. There are some problem associated with that. That is, how do they recognize or how do they go to specific sites? Because it seems that most of the if you look at the diverse genes, all of them are can be, or some of them can be targeted by PCG proteins, some of them can be targeted by HDAC, and some of them can be targeted by DNMT1. However, you do need some kind of a scaffold or an adapter that can take them to the specific site. That is where the new family of RNAs come into play. That is what has been recently known as that link RNAs actually regulate most of this epigenetic program. If you look at the epigenetics, the critical role of the epigenetics, you can see that this epigenetic modulation is involved in almost all the different hallmarks of cancer. So I think it is suffice to say that it is most critical pathway in addition to genome in defining the fate of the cancer genesis, growth, as well as progression. So when you start looking at them, you do look at, with more interest in looking at, whether any of these complex, readers, writers, or erasers can be targeted for therapy. So it is becoming more and more critical to understand the biology of, the epigenetics, the cancer, different cancers, so that, we will be able to be in a better position to target and, or to strategize and target, specific epigenome of the cancer. So one of the things what is known is that, as I was mentioning, you do need some kind of scaffold to take these adaptive proteins to or regulator protein to specific sites. In fact, what has been identified recently is the family of non-coding RNA, that is long non-coding RNAs. They seem to be exactly performing those functions. Here, I have given you, there are some examples. That is, for example, hot air and cyst through the polycomb repressive complex 2 protein, they can methylate histone on specific target gene, promoters. Likewise, you have hot tip that can recruit, the MLL complex to methylation involved in activation. So I think, you can see that link RNAs can, and also this epigenetic, writing can involve both, repression of the gene as well as activation of the genes. In another case, Linko 1268, that activates HDAC-C2, which in turn deactivates and deacylates H3, K9, and K27. That is lysine 9 and lysine 27. That can activate, that can actually induce transcription silencing. In contrast, the link RNA ECCEBPA, that can sequester DNMT to protect the CEBP promoter from DNA methylation. So you can see that you'd be involved in the putting these tags, appropriate tags in the protein histones as well as in the DNA. So I think the role of link RNA is becoming much more critical than what is previously known in terms of epigenetic regulation. So as I was mentioning, I'm going to talk only about the long non-coding RNAs, even the other non-coding RNAs are also involved in epigenetic regulation. So I'm not going to concentrate on that. Primarily, I'm going to look at some of these long non-coding RNAs, that is, 200 nucleotides, more than 200 nucleotides in length, and which activates, and a set of genes or represses set of tumor suppressor genes in the cancer epigenome. So as you know, the link RNAs have, as you probably know, the link RNAs have has been assigned multiple functions. They are both present in cytosol as well as in the nucleus, and they can be involved in several functions. One of the things what we are interested is both what it does in the cytosol. Cytosol, we know that most of the link RNAs can can act as an effector by sequestering some of the microRNAs. And in the nucleus, it can actually, act as a scaffold for several modifiers. It's not necessarily the epigenomic modifier. It's also involved in other modifiers. Today, my talk is primarily on how this can facilitate epigenetic modulation in the genome. So the link RNAs are in different cancers. So I think what we are trying to understand is how these link RNAs differ in each of the epigenome. So what we would like to do is, if you look at the PCG family of protein that is very primarily involved in the epigenetic regulation, that is polycomb family of proteins, they have something like 250 to 300 target proteins. So you need to have a kind of platform so that we can identify what kind of specificity or what kind of specific genes are repressed or activated in specific, cancer or in specific, pathological context in cancer, like invasion or proliferation or metabolic regulation. So we took, we sought to identify this epigenetic regulation primarily in ovarian cancer, because my lab has been, over almost a decade, we have been working on ovarian cancer. And ovarian cancer, probably that it is less than 60% of the patients survive more than 5 years. And early symptoms are not specific to ovarian cancer. So I think it is becoming more and more important to identify the genomic as well as epigenomic targets involved in the genesis as well as progression of ovarian cancer. So, and also there is no effective targeted therapy. So anything you can find out in order to kind of, focus or target the ovarian cancer would be of great help for, most of the patients. Right, currently it is something like 20,000 patients, suffer, almost every year in US alone. So one of the things what we have done is we have done some analysis of, the transcriptome analysis of both coding and non-coding RNA in ovarian cancer, primarily using patient-derived cancer cells. What we observed was there are at least a hub of link RNAs. The link RNAs is given in red, they seem to be regulated most of the coding RNAs. So in fact, we could see these link RNAs are overexpressed. One of them is, PCAT1. You can see that, it is almost, 40%, overexpressed, amplified or overexpressed in ovarian cancer. Likewise, there is another link RNA, which, we have been working on, which is UCA1, that is in almost, 15 to 18%, overexpressed or duplicated in ovarian cancer. Likewise, there is another protein, another link RNA, which is CCAT1. Most interestingly, all these link RNAs have been known to interact with PCG protein. So the problem is this. So you think, you have several link, this is just a limited set. So several of these link RNAs compete with each other to bind, to regulate some genes through the interaction with BCG1. So one of our goal was to identify, first, to develop a platform so that we can identify how, each of these link RNAs regulate, if possible, regulate different set of PCG targeted protein, and then see whether, these targets can be used for, adjuvant therapy in ovarian cancer. Today, I'm going to talk a little bit about, both in our, effort. So if you can see, I'm going to take the example of UCA1. So what you can do is, whether you can identify whether the UCA1 interact with PCG proteins. So in fact, we did some, cross-linking experiment. Also, I think, we have developed an aptamer-based expression of UCA1 in the ovarian cancer cells, and then, precipitate with, the specific, the PRC1 and PRC2 complex proteins, and see whether the link RNA comes with that. And you can see that the link RNA is enriched in all of this immunoprecipitate using, the different, the PCG protein, suggesting that they do interact with PCG protein in the cell. Then, what we want to know was how do they regulate the different, the genes. So in fact, it is known that, link RNAs can act both in cis, position, as well as, they can act in, trans position. That is, UCA1, in this case of UCA1, that is present in chromosome 19. So what it can do is it can actually, provide a scaffold for genes. What we are interested in, tumor suppressor genes that are regulated by PCG proteins, cis position in the chromosome where it is located itself, or it can move to other, it can come out of this chromosome, and in fact where it is transcribed, and it can go and regulate other genes in a transposition in other chromosomal locations as well. So in the first case, what we want to do was we want to scan what all the different tumor suppressors, candidate tumor suppressors that are present in the chromosome 19. We had a list of genes. There are at least 12 different tumor suppressors present in the chromosome 19. When we checked it in the cis position, what we've observed was it is very specific in regulating a set of tumor suppressors. Okay, that is, for example, XRCC1, which is involved in DNA repair, and BCL2, L12, and BCL3 and BAX protein. These are all, anti-apoptotic protein. So when we knock down the UCA1 in these cells, these proteins, usually repressed by UCA1, they show increased expression, suggesting that this UCA1, in fact, acts in cis, regulating these proteins. However, if you look at the cancer cells, it is not a single pathway. It's not a single mechanism that regulates the cancer genesis and progression. You have a coordinated expression of a vast array of genes. So what we decided was we want to look at whether it is regulating other genes, look at it in other chromosome in trans mode of action. In order to do that, what we decided was we looked at whether we can so it can act in two positions, whether we can develop a platform to identify this. What we decided was we decided to construct an antibody array to interrogate the PCG target proteins. Here I have given the kind of, the principle behind that. What you do is, you have an antibody attached to array slide, and then, you have a lysate with, bioaccumulated protein. You label the protein in the lysate, then, incubate that with the antibody array, and then read that in, and wash it off, and then read the version of the antibody and read it in a reader. And finally, you can, see whether, you can identify the proteins that is regulated or that is captured by these antibodies. Okay, so that is the idea. So what we decided was, we looked at the literature and we found that there are 250 polycomb group of proteins, that is, 200 genes that can be targeted by the PRC1 and PRC2 complex. So we obtained this, antibodies from commercial vendors. We optimize, we kind of, dilution, most of standardization we did in our lab. Then we finally, we construct an antibody containing all these, 250 target genes in the antibody array, slide. So after that, we subjected this antibody array analysis using the ovarian cancer cell UCAR8, in which we knocked down the UCA1 and with the appropriate control. When we did that, what we observed was, so there's a kind of typical the heat map what we get. So you can see there are a lot of changes, and this is the kind of the genes that seems to be upregulated or downregulated. What it means is that when you have a signal in the antibody array, what it means is that these signals represent the proteins that are being repressed by UCA1 in its wild type form. When it is knocked down, these genes are re-repressed and re-expressed, and they can bind to the antibody so that you can identify that as a positive signal. So we can see a slew of protein, that's it, the protein that is repressed by UCA1 in these cells. And please remember that all these proteins are, all these genes that are coding these proteins are the PCG targeted proteins. Primarily they belong to the group of tumor suppressors. Some are established tumor suppressors, some are tentatively known as tumor suppressors. Having done that, we then started characterizing them. This is the kind of scatter plot. You can show that we can show that there are multiple proteins that seems to be upregulated, and there are some which goes down with UCA1 when it is knocked down. One can see that again we can add more stringency so that we can define, have absolute confidence in the data we have. And once we have that, we'll tabulated these proteins. You can see that these 18 proteins, 18 PCG target proteins, seems to be very much repressed by UCA1 in ovarian cancer cells. So you can see that most of these things are known to be not necessarily in ovarian cancer, but in different cancers known to be tumor suppressors and targeted by PCG complex in repressing them during cancer genesis as well as progression. Once we had this data, then we wanted to look at I'm sorry, this is not the slide. Then we wanted to look at the biological process that is regulated by this protein using cytoscape analysis as well as string analysis. So you can see most of them activate, most of them are involved in different pathways, including the signaling as well as metabolic processes, which we are very much interested, because we have shown in the past that the UCA1 is also involved in metabolic regulation in ovarian cancer cells. Then we looked at the molecular function. You can see that they are involved in the regulation, because I think all these proteins define so many different functions in the cell, including specific transcription regulator activity, which is known for some of these polycomb repressor proteins. And then we also looked at the pathway analysis. Pathway analysis, we can show that it is TGF-beta pathway, signaling pathway is very much affected, which is known to be one major pathway in EMT as well as cancer cell invasion, even in the case of ovarian cancer. And P53 pathway, which is known to be a major pathway in ovarian cancer cells, that forms a predominant set of proteins that are being regulated by UCA1 via this PCG complex. Then we looked at the protein class, you can see that, some of them are scaffold, some of them are gene-specific transcriptional. So the transcriptional activity seems to be playing a major role. After this, we did a string protein interaction analysis so that we can construct a reactome. So if you look at this, what we can see is that major pathway cluster involves apoptotic signaling. So it is, these proteins that is regulated by UCA1 seems to be involved in anti-apoptosis, and UCA1 seem to repress them so that, the cells can evade the apoptotic signaling. Then we also see a signal that is indicative of operational in terms of hyposignaling as well as notch signaling. There are some evidence that, hyposignaling as well as notch signaling is, present or it plays a dominant role in some subtypes of ovarian cancer. So we think that, this is representative of that. So we are, very much interested in pursuing them. After that, what we did was, we do a limited, characterization. Since I don't think, I have more time, I will quickly go over that. And that is, we looked at the pathway, hippo pathway. We can see that, hippo pathway, the UCA1 is upregulated in almost all of the cancer cell lines we have tested. And concomitantly, we can also see that, the YAP1, which is, indicative of hyposignaling, is also upregulated in these cells, suggesting that this is one mechanism which you are pursuing now to define UCA1 PCG complex in terms of regulating the ovarian cancer cell, ovarian cancer progression. Then what we also wanted to do was how we can use this for therapeutic purpose. So there are several epigenome targeting drugs have been, either approved or being, used in clinical trial. The major problem with them is they are effective only for short term. So one of the things we wanted to test was whether, first, we want to show whether they are effective in a in vitro or ex vivo model. So we can show that, when we use one of these two drugs, which inhibit EZH2, which is, PRC2 complex protein, we can show that GSK 126 and 343 can really inhibit fibronectin, vimentin, as well as twist, which is indicative of EMT and, cancer cell invasion. We have some data on stem cell also. I'm not, going to present it at this point because it is too preliminary at this point. So what we wanted to do was to see whether we can utilize this in combination with the link RNA studies, where we have used in the past that by knocking down the link RNA, we can decrease the tumor volumes as well as tumor burden in the ovarian cancer. This is, indicative of the, silencing of the, UCA1 in this xenograft model. So what we would like to do is whether, we can do this, co-therapy, using the targeting the, epigenome, using this, some of these PRC1, PRC2 targeting drugs, as well as along with, UCA1 silencing, so that, we can have maximal efficiency and, verify the toxicity in terms of, how it affects, small animal model in the lab. So these are all being, the things that are going on in the lab. And probably we'll be able to update probably in our next seminar series. So with that, I'll finish my talk. So what we have done here is we have given you a platform. So right now we have this platform using this antibody array where we can use any of the lysates, any link RNA, to identify what set of PCG targets they are involved. So right now we have used UCA1. Now, we can validate this for other link RNAs as well. So I think, this has proved to be a great, advantage or, a technological tool for us where we can cancer specific as well as, link RNA specific regulation of PCG proteins. And this is, accomplishment in the lab. And then, we have also identified the tumor suppressor genes that is involved in notch as well as hyposignaling pathways, as well as, pro-apoptotic genes in ovarian cancer using their methods. We also have shown that the epigenetic basis for the activation of notch and hyposignaling pathways in ovarian cancer. For example, even though it is known that, notch and hyposignaling is activated in subtypes of ovarian cancer, the basis was not known. So what we are showing here is a glimpse, through which UCA1 can upregulate these pathways through epigenomic, regulation. In addition, we are showing as a proof of concept that, we can use some of these inhibitors. In our case, we use EZH2 inhibitors, and we can use some of these inhibitors to attenuate the cancer specific. In this here, we showed EMT pathway. We are still assessing whether more efficient. Like for example, cisplatin resistant ovarian cancer. It is known that it is epigenetically regulated. So we would like to see whether one can use these inhibitors for that. Then this antibody platform, that is a major combination we have done. And we have also shown that UCA1 is one of the major modulator in ovarian cancer epigenome. And with that, I will end my talk. And I want to acknowledge the MDPA for giving this opportunity. And the work is done by Jihi, Ha, and Ranga, Murali there, Mingda, and Rohin in my lab. And we have a robust collaboration with our clinical scientists, Catherine and Coming5. And all along the project, I could not have been able to do all these things without my, collaborators in Italy. Ciro is my co-chair, and we have been doing so many different things together. This is one of the example of that, and also Yang Sang Song, who is in Seoul National University. With him also, we are doing several of these studies. So these are all part of, our collaborative consortium, which has completed the study. With this, I will, stop my talk, and then, I'll acknowledge my, funding sources for this study.
Thank you so much, Danny, for your talk. So the Q&A session is now open. So if anyone has any questions, you can post them in the Q&A box at the bottom of your screen, or you can raise your virtual hand and I can give you the microphone. So we might have to wait a few seconds for people to ask their questions. Okay, so I see Mohammed has a question. Mohammed, you should be able to switch your microphone on.
Hello.
Hi. Hello. Yeah.
Thank you for the nice presentation. I really have the question to ask about how did you define your 250 genes of polycomb targeted genes? Did you define them based on cancer type, database, stem cells, or do you have a specific approach to define them?
No, actually, this is based on a database. Actually, there is a beautiful, data set is available, identifying all the PCG protein, both in human as well as in yeast. And what we did was, we kind of looked at the homologs, and then looked at the human, the data set, and then we combined together 250 different PCG protein, and obtain the antibodies for them.
So do you expect the behavior of these genes when you note down MUC1 to be different between different cancers, or to behave the same?
Exactly, that's what we are thinking. What we are thinking is this cancer epigenome is different. It is context specific. In different cancers, it can vary. And also different, see, for example, we can show that the pathways which is regulated by UCA1 seem to be, a specific set. In the case of, we see that, PCAT is also upregulated in ovarian cancer cells. That probably, we haven't done that, antibody array yet. What we are thinking is that may regulate different set of, genes that is in one cancer itself. And also in different cancers, for example, in pancreatic cancer, we have different link RNAs that seems to be, upregulated. So we think that, that could be involving, different, set of link RNAs. We want to compile it so that we can kind of, define the plasticity of the epigenome in a context specific as well as cancer specific manner.
Yeah, great. And then if you look at the array, not all genes have been reactivated. Some of them, I mean, the signal was already, for some of them, is visible at control knockdown, while increased at the knockdown itself. But some still the same. So that means to me, I mean, there has been some debate that the PRC2 does not completely repress gene expression, but dampen it. And then I'm just wondering those genes, and that's based on the genomic neighborhoods where the gene is lying, which part of the genome. Is it close to a PRC2, PRC1 region, or it's just closer to an active region? So what I mean is that not all genes get, based on the antibody pull down or antibody array, were reactivated the same. Do you think that this is related to other epigenetic mechanisms? Some genes are more repressed by DNA methylation, for example,
Exactly.
PRC2?
Yeah, exactly. That's what we are thinking. What we are thinking is there are at least, 3 sets of pathways that may be operational. One is, the one of them is PCG. And again, there is a question of, the gene neighborhood. We would, physically one would think that, the gene neighborhood, matter a lot in terms of, the link RNA as well as the PCG protein. So I think, that may, tell something more. And also when you have a collection of co-expression of, link RNAs, we are not sure how that dynamics will work in terms of, activating. So the PCG1 is one mechanism through which, the link RNAs operate. There are, several other mechanisms through which, the link RNAs regulate epigenome as well. So we are kind of cognizant of this, caveats. But one of the things what we would like to see is whether we can get a kind of, outline of the epigenome, context-specific epigenome. Then we can go from there, teasing out the finer mechanism.
Yeah, it would be interesting to see if polycomb localizes to some other regions when you take away one long non-coding RNA.
Yeah, exactly.
Yeah. Thank you very much.
Thank you for your question. Yeah.
Okay, thank you. I can't see any other questions and no hand raised. So, Ciro, I'll let you.
Thanks. Of course, we are collaborating, but I am missing one point. The story of the xenograft where you have the silencing of UCE1, would you expect then that because of that, the growth of the tumor is reduced because you are switching off the Hippo pathway, perhaps, or because of the anti-apoptotic genes are now, what is happening, you think?
Exactly. I think, see, we were not aware of this. That data was earlier than, earlier than what these new findings we have. So now we know, we are wondering, what is actually happening is either because of anti-apoptotic or, either, the, notch or, the Hippo pathway, because, the xenograft involves prokaryote cell itself, which we have used for this antibody array. So I think, we have to test that. And also we have to do, like Ahmed was mentioning, we had to kind of characterize with, other panel of other ovarian cancer cell itself. But I do agree that, that may be the endpoint, which we have missed in the previous study. But I think, we will analyze that now.
I have one more question, Danny.
Okay, well,
this long non-coding, sometimes it acts as a to support the protein structure, the complex structure.
Yeah.
When you knock down it, when you took MUC1, do you see degradation of PRC2 subunits, or they are just staying the same? There's no change in the level of proteins of the PRC2 complex.
That is a good question. We didn't look at it. In fact, we have data now. We can look at it. We have the complete transcriptome, coding as well as non-coding at a given time. We didn't look at it. That's a great point. We'll look into it right away.
And the histone mark, for example, too. If the histone mark goes down on these genes too.
Yeah, we should be. Yeah, we will. It's an excellent point. We didn't look at it. We'll look into it. See, to be honest with you, this is the first time we are presenting our result. Outside the lab. So I think some of these questions we didn't really interrogate in our studies. So we'll start looking at it. Especially this point is very much valid, like your previous observation about the context which you are interrogating this. We'll do that. Thanks.
Yep.
Okay, so I think now we can move on. Thank you very much, Danny, for your presentation and your time. Ciro, the floor is yours.
Yes, thank you. It's now time to introduce the next speaker. And the next speaker is coming from Besançon. I know very well this beautiful town because I'm visiting professor there and honorary professor there. And I was there in February and I was supposed to stay just 1 month, but then I stayed 3 months because of the COVID. So Eric comes from Besançon, from a group working on, as you can see, epigenetic regulation and involvement in EMT, autophagy, and so on. And today he's going to present his data on this epigenetic signature during epithelial to mesenchymal transition in non-small cell lung carcinoma. So Eric, please, and thanks for coming.
Thank you very much for the introduction. So first, I will thank the journal Cancers and Professor Ciro Isidoro for the invitation. All the data I will present to you today have been supervised by me and by Dr. Paul Peixoto, who are working in the INSERM Institute in Besançon, in France, in the University of Bourgogne-Franche-Comté, as Ciro said, in the team right. And we are both attached to the Epigenetic platform. Most of the data presented here belongs to the PhD thesis from Camille Laja. I will speak today about EMT. So EMT for epithelial mesenchymal transition, a phenomenon linked to metastases in cancer and controlled by tumoral microenvironment. An epithelial cell, as you can see here, can undergo reversible phenotypal modification in different and progressive mesenchymal states. And EMT is accompanied by a loss of junction between epithelial cells and a loss of apicobasal polarity in benefits of front-back polarity. EMT is also associated with a loss of expression of epithelial markers and again of expression of mesenchymal markers such as transcriptional factor associated with EMT such as ZIP family, twist, or SNEL. And finally, it is known that EMT is controlled by epigenetics. As already said, EMT is thinly controlled by tumoral microenvironment, and notably by molecules that are produced by immune cells near the tumor. Among these molecules, two sequences, TNF-alpha and TGF-beta, appear the ones with the most efficacy to induce EMT. In the lab, we use the A549 cell model, a non-small cell lung carcinoma cell model, to induce EMT using a co-treatment with TGF-beta and TNF-alpha. As you can see here, using classical microscopy, here with cytoskeleton staining, that means phyloidine-rhodamine staining for actin. I will try to make, I will make a long story short. 2 years ago, we published an article saying that the expression of the immune checkpoint inhibitor, PD-L1, was controlled by EMT. As you can see here, using immunostaining staining, you can see that the expression of PD-L1 is strongly increased in non-small cell lung carcinoma tissue in EMT positive tissue compared to EMT negative tissue. And we confirm this observation in our cell model, which is which you could induce in EMT using the co-treatment with TGF-beta, TNF-alpha. As you can see here, TGF-beta was responsible of the inhibition of the expression of the DNA methyltransferase DNMT1 that led to a global DNA hypomethylation and a local DNA hypomethylation on the PDL1 promoter. In the same time, the other cytokine, TNF-alpha, activated the AKK epsilon NF-kappa B pathway that lead to the stimulation of the transcription of the demethylated PDL1 promoter and its overexpression that contributed to immune escape in these tumors. So, epigenetics, that means here in this paper, DNA methylation control immune escape during EMT. We next analyzed in a paper which was published last year the role of histone post-transcranial modification in EMT. For this, we used 3 different cell lines, 8CHN, A549 cells, and the MCF10A cells, which all 3 could be induced in EMT with the co-treatment with TGF-beta, TNF-alpha. Using transcriptome analysis, as you can see here, or with Venn diagram or oligodendrogram here, you can see that most of the EMT programs were cell line dependent. However, few dozens of genes were commonly regulated, and we focused on these specific genes that could be important for EMT in different cancers. And we also focused on a specific histone repressive mark, which is the 3-methylation of the lysine 27 of the histone H3, as you can see here, which is a repressive mark of transcription. In our A549 cell model, we saw, using the EMT induction with the both cytokine TGF-beta, TNF-alpha, that this repressive mark accumulated in cells. This result suggested that this repressive mark could be important for EMT. Moreover, we performed ChIP-seq analysis, and we observed that most of the genes we were commonly regulated between our 3 cell lines during EMT were controlled by this repressive mark. For example, as you can see here, we observed using QRT-PCR that the expression of EDEM19, MMP9 were induced following EMT induction. On the opposite, the expression of FCNN1A was silenced. The expression of these genes were inversely correlated to the status of the H3K27ME3 mark on the promoter of these genes. As you can see here, we observed a loss of this repressive mark on the promoters of EDEM19 and MMP9, and an accumulation of this repressive mark on the promoter of FCNN1A. So we next focused on the enzyme controlling this mark, and I will thank Danny for introducing PCG proteins in the talk before. Indeed, EZH2 is a histone methyltransferase, which is able to catalyze the methylation of the lysine 27 of the histone H3. And on the opposite, KDM6B, which is also called GMGD3, is a histone demethylase, which could remove this mark. We recently reviewed the role of this enzyme in cancer during EMT in the journal Epigenomes. And for example, we observed, we reported in this review that the expression of EZH2 is frequently shown to be overexpressed in many different cancers, and sometimes the expression of EZH2 has also been correlated with aggressiveness, invasion, and EMT. Similarly, the expression of KDM6B has been also associated with different cancers, but this enzyme is less studied than EZH2. We recently, this month, published an article in Cancers focusing on the role of EZH2 and also KDM6B in EMT in non-small cell lung carcinoma. And our first result was to show, using immunohistochemical staining, that the expression of KDM6B, but also EZH2, were overexpressed in non-small cell lung carcinoma EMT positive tumors compared to EMT negative tumor, as you can better see here using quantification. And interestingly, the expression of EZH2 on KDM6B were correlated. So this data suggested that this both enzyme and not only EZH2 were important for EMT in non-small cell lung carcinoma. So we decided to modulate the expression of these genes in our I549 cell model. For this, we use a transduction model to obtain stable cell lines. As you can see here, stable cell line expressing EZH2, and here, stable cell line expressing KDM6B. And we observe using classical microscopy or actin staining using phalloidinodamine staining that we observe some mesenchymal-like phenotype. That means here an increase of cell length, for example. This was confirmed at the molecular level using, for example, QRT-PCR experiments. We confirm that the overexpression of EZH2 and also KDM6B increased vimentin expression, MMP2, metalloprotease, ADAM19, and on the opposite, the overexpression of EZH2 and KDM6B silence the expression of the gene CDH1 coding a cadherin, a protein which is important for, which is an epithelial marker. Finally, we wanted to know if the modulation of overexpression of this protein was important for EMT phenotype. So we quantify cell invasion using Trans12, and we observe that the overexpression of EZH2 and also KDM6B seems to promote cell invasion. So overexpression of EZH2 and KDM6B appears to promote UMT-related phenotypes. So we, on the opposite, decided to inhibit the catalytic activities of this enzyme using selective inhibitors. Danny already spoke about this one. So we used the GSK343 inhibitor to inhibit the expression of EZH2 and the GSK-G4 for the inhibition of the activity of KDM6B. Interestingly, we observed that the chemical inhibition of EZH2 or KDM6B also promoted EMT-related phenotype. That means something similar that we observe when we overexpress the two proteins. Indeed, as you can see here, using actin staining, we observe during the EMT induction using the TGF-beta, TNF-alpha co-treatment, we observe an increased cell length. That means mesenchymal-like phenotype. This was confirmed at the molecular level using QRT-PCR, immunofluorescence, or Western blotting experiments. As you can see here, we observed a decrease, which was more important, of the expression of CDH1, an epithelial marker, when the catalytic activity of this enzyme was inhibited during EMT. And on the opposite, we observe an increase of the expression of the vimentin or N-cadherin markers during EMT when the catalytic activities of this enzyme were inhibited. Very rapidly, we confirm this data using CRISPR-Cas9 models in one clone presenting the inhibition of the expression of EZH2, and in one clone presenting the inhibition of the expression of KDM6B. We confirm the increase of the expression of vimentin. So, to result this apparent paradox, we hypothesized that EZH2 and KDM6B could target different genes during EMT. So for this, we performed some ChIP-seq experiment against the H3K27M3 mark, or against the protein EZH2, or against the protein KDM6B. And we confirmed that our hypothesis was correct, since only 570 loci were common between this enzyme, suggesting that this EZH2 and KDM6B target different genes during EMT. We next crossed this ChIP-seq data with previous transcriptome data we obtained in the lab to obtain some list of genes that could be regulated by this enzyme during EMT. And we obtained a list of 168 genes that could be putatively inhibited by EZH2, and a list of 560 genes that could be putatively activated by KDM6B. We focused on few of these genes in order to try to confirm this list and to see if some of the genes could be important for EMT. We chose the Win5B gene, ADAMTS6, and INHBB. And we confirm using QRT-PCR experiment that the expression of Win5B and ADAMTS6 were increased during EMT. On the opposite, the expression of INHBB was silenced. And we also confirm that these expressions were inversely correlated to the status of the presence of the repressive mark H3K27ME3 on the promoter. As you can see here, we observe a decrease of the presence of this repressive mark on the promoter of Win5B and also ADAMTS6 during EMT, and an accumulation of this repressive mark on the promoter of INHBB. So we suggested that these genes could be new biomarkers of EMT that could be regulated by epigenetics. And for this, to confirm this, we performed some immunohistochemical experiment in non-small cell lung carcinoma. As you can see here, easier to understand using quantification at the bottom, you can see that we confirm that the expression of Win5B and also ADAMTS6 was increased in non-small cell lung carcinoma EMT positive tumor compared to EMT negative tumors. On the opposite, INHBB expression was partially silenced in EMT positive tumor compared to EMT negative tumor. Finally, we asked whether these genes could be important individually for EMT. So we decided to inhibit their expression using RNA interference. As you can see here, the inhibition of the expression of NHBB, of Win5B, and a partial inhibition of ADAMTS6. And we perform cell invasion assay. As you can see here, the inhibition of the expression of NHBB seems to promote cell invasion, as expected. On the opposite, during EMT induction, we observe that the inhibition of the expression of Win5B and also ADAMTS6 inhibited cell invasion. So this data suggested that this three genes could be important for EMT and are regulated by epigenetics. I will really thank Professor Ciro, Isidoro, and the journal Cancers for the invitation. And we thank also all the people who were involved in the production of the data. And maybe I could say you to save these dates next spring for EPI meeting and a face-to-face meeting in 2022 in Besançon. Thank you very much.
Thank you, Eric, for your presentation. We already have one question in the Q&A box from Cadles, who has asked, how should we interpret increased PD-L1 expression upon EMT? Protection about chance attack of migrating normal cells from immune system. And he apologizes if he misunderstood your point.
Yes, we suggest that the PD-L1 expression, which was induced during EMT, could contribute to immune escape in this tumor, and this could be controlled by epigenetics. So we suggested that the tumoral microenvironment was very important to control epigenetic status. In this case, that means DNA methylation near in the immune cells which were near the tumor, and the expression was also controlled by the presence of the cytokine which were produced by the immune themselves.
Okay, thank you. Mohammed has asked a question, so I'll just allow him to talk. You should be able to switch your microphone. There we go.
Thank you. Have you found PD-L1 expression to change when you overexpress EZH2 or KDMD6B? Is PD-L1 expression EZH2 KMD6B related or not?
Excellent question, but we didn't try to see that. So probably we will do it, but we did not actually.
I really think this is really interesting and very exciting that when you have the methyltransferase and the demethylase overexpressed at the same time. And I really think that maybe it's worth looking at the nuclear periphery where they have been recently, people looked at like such, maybe KDM6B is more localized to areas of the nucleus close to the nuclear pool complex that will allow transcription and export of the transcripts, while PRC2 is more into the inside of the nucleus. And that may explain why you have both active methyltransferase and demethylase high in the same cell.
Maybe. We observe that EZH2 is present not only in the nucleus, but also in cytosol. So probably there are regulation of cytosolic protein that could
exactly.
then control nuclear protein. So it's complicated. Our hypothesis, which we are now testing, is that this protein could interact with specific other proteins that could be targeted on specific promoters. So we are now searching for partners of this enzyme that could be different during EMT. It's our hypothesis now.
No, thank you.
Thank you.
Thank you for that. I can't see, there don't seem to be any more questions. Ciro, oh, sorry. You're still muted.
Okay, thank you, Charlotte. Oh, Eric, very interesting. And by the way, you will say hello to Regis and all my friends over there.
Of course.
It seems to be redundancy in the signaling and so on. So have you tried to put together the two inhibitors?
Yeah, but we did not perform a lot of experiment with both. When we analyze the status of the H3K27M3 mark, we observe a decrease of this mark, as if it was only with a single treatment with the EZH2 inhibitor. We did not perform ChIP-seq experiment with both inhibitors, so just preliminary results.
Okay, one more point. And what about the reverse of the, I'm thinking of mesenchymal to epithelial transition.
Yeah.
What happens there?
We tried on this on the first paper, so we know that if we remove the cytokine TGF-beta on TNF-alpha, the cell will reverse on epithelial cell in about 5 to 10 days, and we will have a decrease of expression of PD-L1, but we did not perform this experiment for the paper concerning the roles of EZH2 or KDM6B.
Okay, thank you.
Thank you.
We have one more question, Mohammed.
Yeah, hi. Sorry. So does your data mean that you cannot give EZH2 inhibitors or KDM6B inhibitors to patients or metastatic patients in that case?
It was a surprise for us. So that's why we are trying to better understand what can trigger this enzyme on specific promoters, because it clearly appears that inhibition of EZH2 or also inhibition of KDM6B are not good in this kind of cancer. Probably we suppose that we hypothesize that there is a common regulation between these two enzymes, and maybe we can imagine a balance between H3K 27 ME3 methylation if we have a lot of EZH2 or a lot of KDM6B or few EZH2 or few KDM6B, something like that is modified when you are targeting one, you will have probably a response concerning the other. That's why probably when we use an inhibitor of EZH2 or also KDM6B, we have a similar phenotype. So probably it's not a good idea to use these inhibitors in these cancers.
I agree with you. I'm just, because most of the data is based on cell line work, I guess. I mean, are you planning or do you think of using this or doing this in vivo?
We just began to do some in vivo experiment with not favorment for with the inhibitors, but
I understand,
for the overexpression models.
Just to study the M-base and metastasis issue and the stuff like. Thank you.
Thank you.
Okay, thank you very much. I see no more questions, so I guess thank you, Eric, for your time and your presentation, and I hand the floor back to Ciro.
Thank you.
Thank you.
Okay. So next speaker, it's a big pleasure now to move to Umber Cheema. She's an expert in 3D tumoroids, and this is a very interesting way to model the cancer organoids and the tumor microenvironment. And she did a great job, great work to develop a way to set this model with humanized in vitro, humanized cell lines, cancer cells. And so she's going to show us how it works, the model, to interrogate the tumor stroma boundary. Thanks for coming, Umber, and it is now your turn.
Thank you very much, Ciro. I'm just going to share screen. I hope it's clear. Thank you so much for the invitation, and the two talks have been fascinating, so I'm looking forward to telling you all about what we do. So my talk is going to be a little bit different to what you've heard so far, and I'm going to specifically, as Ciro has said, focus on bioengineering 3D tumoroids. And the reason why we think this is so important for the study of epigenetics and other cellular signaling is because by generating controlled 3D in vitro models, you can really interrogate specific types of cell signaling and changes, for example, in methylation patterns between different cells, and specifically in cancer. And really what I'm going to focus on, spending quite a lot of time today, is about focusing on how we can engineer the stromal compartment, or the tumor microenvironment in particular. So the field of bioengineering and tissue engineering is a multidisciplinary field. So it's not only just about culturing cells in a 3D environment, but it's about trying to recapitulate all the aspects, all the biophysical aspects which can control that cancer and its growth in its cells. So for example, you can look at chemical cues in terms of growth factors and cytokines. You can look at further biophysical parameters such as geometry, stiffness, hypoxia. All of these things play a role in cell functions, and specifically in cancer function as well. Not only that, but when you engineer a tissue or a 3D in vitro model, it's always worthwhile putting it into perspective that tissues in the body are complex. There are vascular networks, neural networks, immune component, and without each of those levels, each of those systems filtering into your models, you don't necessarily get something which is biomimetic. And that's really been the focus of a lot of the work that I have been doing with my team and with other collaborators. So if we take just like a quick snapshot of a solid tumor, the solid tumor is cancer cells which are growing in an unregulated manner. They are not necessarily in any kind of tissue with any kind of architecture, but they are proliferating at a very fast rate, they're depositing matrix, and they're forming stiff tumors. But the cancer cells within that tumor are interacting with their stroma, with their native environment which surrounds them. So they are interacting with those cells, they're converting those cells. They're also forming chemotactic gradients of angiogenic growth factors because they're trying to recruit the host vasculature as well. So the models that we develop try to recapitulate this complexity so that one by one we can dissect out each interaction those cancer cells have with each component part of the tissue. Now, I always do a slide on collagen, collagen type 1 specifically, because it's the matrix of choice that we have for generating and building our tumoroid models. So collagen is the predominant extracellular matrix of most tissues in your body. It's widely used in 3D culture and tissue engineering, which means there's a lot known about how cells interact in that environment and how they are altered by the environment. And there are three main ways in which we can control collagen architecture. The first is through direction or orientation. So we know that many tissues normal stromal tissues are highly orientated and organized. And we can use techniques to try to do this with the tumoroids that we build. The second is density. So we know that density is important, particularly where we're talking about stiff cancers, because low density hydrogels are the most commonly used tissue engineering scaffold, but they really are less than 1% collagen and they're mainly water. So what we try and do is use techniques to increase the density to make it more biomimetic to tissues within the body. And then the third and final aspect is collagen fibril diameter, which we are able to control, but we've not yet introduced into our tumoroid cultures. So this is just a quick overview and a schematic of the kind of tumoroids that we have been generating within our lab. And I've just got pictures of two of the PhD students, Tarek Magdeldin and Judith Pape, who have been working on this. So what we essentially try to do with our model is we generate artificial cancer masses and we implant these or embed these within engineered stroma, which are vascularized. And I've got a picture on the right-hand side which is showing you a 24-well plate. So you can see how consistently we're able to produce these cancer masses and implant them in 3D within our stromal surrounds. The really nice thing here is that you can specify the matrix and cellular composition of each of your component parts, and you can have a very clear boundary between your tumor or your cancer and your stroma, and you can specifically interrogate that. And really that's one of the benefits that we see with tumoroids, this concept of a multi-compartment model where you can dissect out specific cell populations. So just in the context of the type of models which exist, and many of you will be familiar with these, we have spheroid and hanging drop, we have matrigel, organoid and organ on a chip, we then have tumoroids, and these are all in the context of having in vivo models and PDX models. Now, we believe that the major benefit of the tumoroids is to do with the stiffness that we're able to achieve using a technique called plastic compression. So as I was mentioning earlier, the majority of people do use collagen hydrogels, which when you look at their published stiffness rates, they're about 330 pascals, and they can go as low as 100 pascals. Now, in the framework of how the stiffness of a cancer, colon carcinoma tissue, for example, the stiffness of that material is between 4 and 14 kilopascals. So if you're culturing cancer cells and you're trying to derive any kind of meaningful information about them, when you're culturing within hydrogels or even matrigel, which is a great model for getting the cancer cells to grow, what you're not doing is recapitulating the native stiffness that the cancer cells would reside within. But within a tumoroid setting, as long as you apply the plastic compression, and we've done specific tumoroids interrogating the difference between hydrogels and the stiff matrix, you can sit within that stiffness range that cancer cells would normally be exposed to. So in this case, about 7 kilopascals is the range of stiffness that we've been able to look at. So one of the first things we did is we wanted to simply see how cancer cells aggregated and how they invaded where we had a dense cancer mass at this 7 kilopascals stiffness range. And if we embedded these cancer masses into acellular stromas, which were made of very soft collagen hydrogels, we were interested to see what happened to these cells. So this is an exemplar. I'm using 2 cell lines here. So the HT29 is a colorectal cancer cell line, but it's a low invasive cell line. And the HCT116 is a much higher invasive. It's got much higher invasive capability. And what we can see very clearly using just phase contrast microscopy is over time, the cancer cells aggregate to form spheroids or clusters, and they migrate out and they invade out into that stromal compartment. But one of the most interesting things was that where we had a soft hydrogel surround, there was a very distinct difference in the way in which the cancer cells invaded. So for the low invasive cancer cell line, they invaded exclusively as spheroids. And you can see that from the H&E images on the lower part of the panel. Whereas when we took our higher invasive cancer cells, the HCT116, they invaded exclusively as cell sheets. And again, we've confirmed that with the H&E. But what we found is that when we moved to a matrix which was much more representative of the stiffness in a normal cancer, we completely lost this specific type of this morphology difference. And what we found is that both the HCTs 116 and the HT29s were both invading as sheets and as spheroids. And we could clearly use the model to generate very precise quantitative data, both on the distance of invasion and the surface of invasion, where it's very clear to see that the HCT116 were invading greater distances and invading with much greater surface area compared to the lower invasive cell lines. So now that we had kind of established just a baseline of how these cancer cells were invading in soft and stiff collagen gels, we then became very interested in vascularizing the stroma and making it much more biomimetic in terms of from a cellular perspective. So we then went about engineering 3D capillary networks in our stromal compartments. So this was a PhD done quite earlier on by Katerina Stamati. We used human umbilical vein endothelial cells to, our hypothesis was that they would generate kind of vascular networks within 3D. But we found that, again, the physical environment was very important in terms of directing cell behavior. So where we put endothelial cells into a plain collagen hydrogel, irrespective of diffusion, which we have measured, diffusion coefficient, all the endothelial cells would always migrate to the top surface. They would fuse to form a cobblestone sheet, and we have evidence of apical basal polarity, which we saw by specific markers. The only way we could get the cells to fuse in an end-to-end manner and form a kind of microvascular capillary network was through the addition of basement membrane proteins, specifically laminin and collagen type 4, to the collagen matrix. So no growth factors, no other cells, simply basement membrane proteins in our collagen gel, and we got this very nice end-to-end fusion of endothelial cells forming these capillary networks. And on further investigation, we were able to show that the way in which these basement membrane proteins direct the way endothelial cells fuse is to do with the regulation of receptors, or the upregulation of receptors on endothelial cells. So where we had collagen laminin constructs, we put in our endothelial cells, we could clearly see that 15%, up to 15% of the cells were expressing the receptor 2 for VEGF. So the composition of the matrix composition of the tumoroid was able to direct how endothelial cells uptake VEGF. So we then brought all of this together and we generated vascularized stroma and embedded our cancer masses within those stroma. And what you can clearly see here is that with the CK20, that's a cytokeratin 20 stain, so it's staining the cancer cells. You can see that that kind of almost luminescent green cancer mass, which is autofluorescence from the collagen that we have. You can see the outgrowth of cancer cells, and you can even see entire cancer bodies, kind of spheroids or clusters, breaking off and going into the stroma. So essentially behaving a bit like metastatic cancer. What's slightly more interesting is the fact that the vascular network that we formed, which is stained by CD31, aligns completely perpendicular to the boundary of that cancer mass. And we were further able to show that that was due to the formation of chemotactic gradients of angiogenic growth factors being produced by the cancer mass. And when we studied this in depth, we were able to clearly show that the angle of our endothelial structures gradually changed over time as the cancer mass was allowed or generated those angiogenic growth factors. So we saw that from day 7 to day 21, there was a clear shift in the orientation of those endothelial structures so that they were completely perpendicularly aligned to the cancer mass. We then decided that we wanted to make these patient-specific. We wanted to have cells, patient-specific cells, and we also wanted a cancer-associated stroma. So we know that cancer cells interact with stromal cells and are able to influence them and convert them to cancer-associated fibroblasts. So we did that within our model. we flipped from a normal fibroblast cell, a primary cell, to a cancer-associated fibroblast, which was patient-specific. We received numerous patient samples, but I'm just going to focus on the first 3 that we had. They were obviously randomized in terms of they had numbers T7, T10, T11. We received them from mixed genders, from mixed age ranges, and they were mainly adenocarcinoma, although we did have one neuroendocrine tumor. And what we did firstly was every patient sample that we got, we wanted to confirm that they really were CAFs. So we did a Vimentin stain, where we clearly saw Vimentin staining of the cells. We looked at Western blots, specifically for alpha smooth muscle actin, which is known to be increased in CAF cells. And you can see compared to our dermal fibroblasts that we were using, all of our cancer-associated fibroblast populations upregulated alpha smooth muscle actin. And this was also on the backdrop of our cancer cells not producing significant amounts of alpha smooth muscle actin. We also did gene assays, so we looked at ACTA2 expression, and we also tested metabolic activity because we know that CAFs are very metabolically active. So really, once we had confirmed that we had CAFs, and this was done for all of our samples, we added them to our tumoroid model to see how they would alter cancer invasion and vascular network formation. So the first thing we were able to clearly show was that there was an increased invasion in the presence of CAFs. So when you flip your tumoroids to a cancer-associated stroma, you have approximately 10-fold increase in some of the samples of cancer invasion into the stromal compartment. So those CAFs are aiding and abetting almost the cancer to invade into those stromal compartments. What was less interesting, wasn't what we were expecting, is that the presence of CAFs almost completely obliterated the formation of any kind of vascular network, which is not what we were expecting, because we know that CAFs are able to promote cancer survival, and they can do so by recruiting host vasculature as well. So we then decided that this may be due to a difference of a study of vasculogenesis compared to angiogenesis. So because we seed single cell endothelial cells and allow them to form networks, if we introduced a cancer-associated fibroblast on the same day, there was a possibility that the networks weren't able to form in the first place. So we redesigned our experiments. So we had normal tumoroids with non-cancerous stromas forming up until day 21. On day 21, we introduced a CAF component, so the cancer-associated fibroblast, to an already existing vascular network. And then 7 days later, we analyzed those networks to specifically see whether the CAFs had remodeled the matrix in any way. And you can see just from the indication, just the one micrograph, the image that we've got here, that there was significant difference in the complexity of the networks that we formed. So very clearly, the introduction of CAFs at day 21 led to the disruption of the pre-existing endothelial networks. And this was mainly to do with junctions and loops and complexity of the network. So we got a lot more single-stranded kind of vascular networks, which weren't necessarily connected to one another, which may be an indication of how leaky vasculature, which is described in cancer a lot, may come into play. Now I'm going to briefly move over to now a 3D ameloblastoma tumoroid model. So I've talked about quite a basic tumoroid stroma, where we have a population of fibroblasts which can be cancerous or normal, and an endothelial network. But really, we were with this particular work, we were interested in creating a bone stroma. And that's because ameloblastoma is an epithelial tumor, specifically in the jawbone. So what this tumor does is it results in bone resorption, and there is really very little you can do to treat it other than resect out the entire jawbone. So it's very debilitating and disfiguring for patients. And so this is Denise Bakalji, who is just currently finishing off a PhD in this area. We received cell lines from patients with two different subtypes of amyloblastoma. The AM1 is a plexiform amyloblastoma, and the AM3 is a follicular amyloblastoma. But what was very encouraging when we compared histology slides of our tumoroids with patient samples was that they showed the same form of cancer cell morphology and cancer invasion as they would within the patient. So plexiform is known as a subtype which encourages the formation of anastomosing cords of the tumor, and clearly we could see those same anastomosing cords within our tumoroid models. And follicular amyloblastoma is much more to do with the formation of odontogenic islands, which invade in that same using the same morphology. And we clearly saw with the AM3 that they were doing exactly the same. So again, this then gives us confidence that our tumoroid model and the parameters that we've put in place are biomimetic, and they are able to recreate or regenerate the same environment that you see within the human body or tissues within the human body. We then set about tissue engineering bone in 3D, which seems like a lot of people do, but we really wanted to incorporate it within our tumoroid model. So we know that if you take osteoblasts from rat calvarial bone and you seed them on 2D, using just phase contrast microscopy, after a period of 14 days or even 21 days, you see these nodules forming. And by eye, they're visible as white mineralized bone, but using phase contrast microscopy, they're just like black dots that you can see. So we compared the soft collagen hydrogel and the stiff tumoroid gel, and we seeded cells either on top or within the matrix, and we could clearly see that within the tumoroid models, we were able to generate really high levels of bone. We did further characterization using the standard techniques that alizarin read, transmission electron microscopy, height measurement, but probably one of the most interesting things is when we did nano CT, you can clearly see the formation of woven bone deep within these bone nodules, and the height of these bone nodules was also very large. It was 120 microns. Considering tumoroid cultures are about 220, 230 microns in height, they really were very large, and they were visible by eye, so you could see them. So we then wanted to, if I just quickly overview then, so tissue engineering bone in 3D, we were able to do it earlier using our tumoroid model. We did it at a significantly higher level, so there were more bone nodules and they were bigger. But the idea was then to bring in the amyloblastoma tumor and implant it into our tissue engineered bone. So what we did was, when we introduced the amyloblastoma before day 9, we completely inhibited any kind of bone formation. So you can clearly see that on the first panel you have 3D bone forming. Where we've introduced the cancer on day 6, in the phase contrast microscopy, you can see the boundary of the amyloblastoma tumor mass, and you can see the bone formation, the active bone compartment, and you can see there was actually no bone formation. There was evidence of no bone formation. So amyloblastoma specifically was able to stop this kind of bone formation. I should also mention that we also used a comparator cancer cell line. So we used the MG63, an osteosarcoma cell line. And whilst we saw some reduction in bone formation, it didn't obliterate it. So this was very amyloblastoma specific. So the amyloblastoma cells were able to act on the osteoblasts to get them to stop forming bone, which is something that would happen within the patient. So where we introduced the amyloblastoma after day 9, and day 9 became quite critical, although we don't completely inhibit bone nodule formation, we limit the size of the bone nodules formed. So they formed at a much, much slower rate. So even at that stage, amyloblastoma had an effect on this population of osteoblasts. Now, probably just something that I would like to finish off on really is the importance of matrix complexity for tumoroids. And this is an example of an osteosarcoma tumoroid that we've developed. So we developed tumoroids for osteosarcoma, either using the 143B osteosarcoma cell line or the MG63 cell line. And we clearly saw that where we generated more complex stroma, which contained bone components, we were able to enhance the efficacy of the drug doxorubicin in preventing outgrowth of those tumoroids. So it sounds counterintuitive that the more complex you make a stroma, you actually aid drug action. But certainly that's what we've been able to see within the tumoroid models. So if I now just kind of briefly go over some of the take-home messages, I think hopefully this talk illustrates that we can engineer multi-compartment tumoroids with very clear and defined tumor and stroma boundaries that we can interrogate. The stromal matrix density and cellular composition can directly affect cancer invasion. Where we make the tumoroids cancer specific, so they're cancer associated stroma, we then enhance cancer invasion and we disrupt vascular networks. We are able to tissue engineer bone as a stroma, and where we studied amyloblastoma tumors specifically, we were able to prevent osteoblast-driven bone nodule formation. And we strongly feel that tumoroid models can be used to interrogate the tumor epigenetics. And certainly that's an area of work that we're now pursuing because we realize the power of the model. So thank you very much. I think it's just pertinent that I acknowledge all of the funders and all of the people that have helped with the work. So happy to take any questions. Thank you.
Thank you very much, Umber. So you can... So the Q&A session, sorry, is now open. So if you have any questions, please post them in the Q&A box or raise your virtual hand.
Well, I can start while someone else, correct?
Okay, yeah,
Thank you.
please do.
Umber, amazing system, very beautiful model, and lots to do and lots to learn from that.
Yeah.
It's indeed astonishing that the CAFs do disrupt the angiogenesis and the network of. But that is because you added CAFs to the system.
Yes.
I'm wondering what happens if you add normal fibroblasts and then they become CAFs. Can you see that? Of course, I guess.
Yeah, I think the important thing here is I totally agree. What you would want to see is you would want to have in that system cancer cells signaling, converting those normal fibroblasts into CAFs, which ideally is exactly where we wish to go. I think right now we probably can't do that, but it's something that, especially as we start focusing on interrogating specific cell populations, and we're probably going to do that with mass spec now, so we're probably using that technology. It will be interesting to see how those different cell populations interact with one another, but I think we need more technology. So I'm not a molecular biologist by trade, but it's something that I'm working with molecular biologists and people who work in epigenetics to really focus in on that conversation between those different cell populations. So I agree, we're using quite simplified models, but I would still say the simplified models are important for us to build levels of complexity to interrogate those very questions. But yeah, it's a completely fascinating area and it's something we really want to work on. So the communication across that boundary between different cell populations. So yeah, I take that on board.
Yeah.
It's something that we want to work on.
One more question. What would be the ratio between the number of cancer cells and fibroblasts or CAFs you add? And also I could see that the timing you add the CAFs change the things somehow.
Yes.
What is the ratio you start with?
So, I will have to probably double check with the PhD students, but from my understanding, in our tumoroids, we always use about 50,000 cancer cells in our cancer mass. Obviously, as they form spheroids, we understand that the core cells will die because that's, as they grow bigger, that's exactly what the spheroids do. In terms of the fibroblasts, from what I can tell, we were adding 100,000 cells per construct. But you see, once you do the plastic compression, because we increase the density so highly of the collagen that the density goes much higher than 100,000 cells. So absolute cell numbers is less important than concentration, but certainly they're quite high cell numbers. And we probably use in the region of 25 to 50,000 endothelial cells as well. So you're talking...
Sorry, go.
Yeah, no, perfectly happy. Yeah, so they're roughly our numbers. But if you go up to the publications and I check with the PhD students, they'll tell you exact numbers. Yeah.
I will check the numbers. But one more thing. So when you say you are going to increase the pressure, is this because you want to mimic the situation in vivo? Why? I mean,
So,
at that point, do the fibroblasts, are they able to enter the cancer?
yes. So we have clearly seen. So I've shown clear migration of the cancer cells into the stromal compartment. We have less evidence of the fibroblasts going into the cancer. We have less evidence of that, but we do have evidence of endothelial cells migrating through acellular rings. So we've actually developed multiple models. So you can have a cancer mass with an acellular collagen ring and then your stromal compartment. And we clearly see endothelial cells migrating through boundaries to get into the cancer mass. So we have clear evidence of that. It's within the publications that we see. But we have less evidence to show the fibroblasts migrating into the cancer mass.
But that's maybe because you use high density gels, right? Or
yeah, we do. So
full size matter, I guess.
Yes, so so some of the early work I showed you, so the Magdalden paper was using the cancer masses are always dense, they're always stiff, we always represent, but we have stromal compartments which are soft and which are stiff, and we clearly see differences between the migration. However, I will say that when those stiff matrices of collagen, we've done all the studies on diffusion coefficient of oxygen, of glucose, we've looked at migration studies, and we see cells that can migrate through the layers. It is collagen and it's remodeled. So that's what cancer and other cells are really good at doing. They're really good at remodeling collagen and other matrix components. So if you look at NMPs, if you look at TIMPs, if you look at the host of matrix remodeling, you will see high levels of those within cancer. So this work started off non-cancer. So there's a significant body of work using plastic compressed collagen for fibroblasts, for epithelial, other types of epithelial cells, for muscular cells of the musculoskeletal system. But I do agree, it is interesting for us to study the cell infiltration.
Yeah, and then my point is, I have 2 points. I mean, you doing this as a research. I mean, now how easy it's going to be for us as researchers to buy this?
Yes. So actually, the system that we use, the plastic compression, I'm on the original publication in 2005, but it's patented and you can purchase it from Lonza. So it's called the Raft system. I'm sorry I didn't include it on my thing. But if you check any of the publications, you can actually purchase the plastic compression kits from Lonza, and you can do the compression yourself. Certainly, if you looked at the publications, all of the methodologies are there. And also, we're not very exclusive. Very happy to share with people. If you want advice on methodologies, we will very happily either give you the information or collaborate with you.
And then you can actually culture, I hope I did not miss that, but you can culture cells directly from patients using these.
Yes. So I didn't show today. What I showed today was the cancer-associated fibroblasts. The patient-specific cells is really difficult to grow them. We've had success with renal cancers. So renal clear cell carcinoma is our focus and what we've been doing. So we were able to get them from patients, have corresponding cancer-associated fibroblasts, and we were able to grow them in tumoroids. We've not yet published that work, but all the data exists, so we've been able to do it. And now we're moving over to, so for example, for the amyloblastoma, we're going to focus specifically on getting those patient cells in. Growing cancer is difficult. Growing cancer cells has been difficult. We've been doing it for 5 years and we've struggled. And I think we've only knocked the renal on the head, so we're able to grow the renal cells. But the colorectal has been difficult. We now have other technologies and we have other collaborators who are going to help us hopefully do that.
And nothing about hematopoietic bone marrow derived tumors. Have you tried, or do you know anyone who's doing that?
So far, no. Well, that's not an area that I've not necessarily touched. Certainly, we've put in grant applications around it, not successfully funded yet. But yeah, we are interested because we think that with a pulsatile media flow, the hematopoietic specifically cells, we think that we're going to get really good data with that.
And then talking about the doxorubicin data, you said that you got a really nice response by doxorubicin treatment.
Only for the 143Bs we got it.
Yeah, are they sensitive to doxorubicin in culture or in 2D?
Yes.
In
So
2D?
they were sensitive in 2D. We have actually published that as well. But yes, they were sensitive in 2D and only in our complex tumoroids. So where we had simple hydrogels, we did not see response. So as we build in complexity, that's when we get the response.
Okay. So are they more sensitive than 2D? I mean, the concentration wise, are they using or?
No, so we did a full parameter. So we did lots of different types of concentration.
Okay. Okay, because it would be interesting to compare the 2D, the so far accumulated 2D responses to cell lines and patients' cells to your model.
Absolutely. I think that's our aim, and that's exactly what we would like to do.
Yeah.
But as you know, growing cancer cells is difficult. So we're kind of playing catch up with some of our cell line work and our patient cells.
Yeah, and the one thing I'm really interested in is that this hypoxia versus normoxia thing. With the spheroids, I assume that the cells that are in the center of the spheroid are hypoxic. Have you picked these kind of oxygen at the surface and in the middle of the spheroid?
Yeah. So if I were to just say that yes, we have actually published on this. Okay. So we can clearly see that within the core we have lower levels of oxygen compared to the surface. We not only see cell death in the core, but we see movement of cells from the core to the outer periphery of our spheroids and of our entire cancer mass, actually. And I think that's in the 2014 paper. So we used a system of fiber optic probes to specifically interrogate and measure the oxygen in different levels. I do apologize for the noise. That's my children coming home. I'm sorry.
Thank you.
Thanks. We have another question from Pankaj. You can talk now.
Yeah, thank you, madam. Myself, Dr. Pankaj Kumar Singh from IIT BHU. Here, your study on the cancer is very nice. I have also done study on the colon cancer. In that, I want to ask that you have done your work on the cancer, whether you have gone through some use of nanoparticles in your study.
Yeah, so personally, I haven't looked at nanoparticles, but I do have collaborators and people that I work with, mainly Marilyn and Louisa do, Sandy McRobert, they're both professors based at the Royal Free, who do use technologies for delivery of drugs via nanoparticles, and they have used the tumoroid system. They've probably published on that as well, but certainly we collaborate and we work together. I think if you look for Louisa Du and McRobert, you'll be able to find that literature.
Which nanoparticle you have used?
I must admit, I'm not an author on the paper, so I don't know the exact nanoparticle formulation. But certainly, you know what, if you email me, I'll very happily introduce you to them. I will send you their paper. But I know that they have published and they've been working on that area.
Okay, thank you.
Thank you. We have a few questions in the Q&A box. So one is Andreas, who said, very interesting model. Thank you very much for this presentation. My question would aim at a little bit different technical aspect. Can you also address perfusion, for example, for compound delivery to your tumoroids?
So personally, I haven't introduced a perfusive, a pulsatile media flow system. That's something that we're interested in doing. But certainly in terms of compound delivery for drugs, it's something that we look at. So we do measure efficacy. We do look at how the drug penetrates through, but probably the preliminary data based on diffusion of things like glucose and oxygen and lactate, we have published within collagen. So we do know that molecules of certain sizes will travel through, and we know what the diffusion coefficient is. So that is specifically the work. There is more that needs to be done, certainly on the perfusion, because we want to have a pulsatile media flow. But that is something that we are working on currently.
Okay, and then Gloria has asked, can cells, i.e. tumor or CAF be isolated from your culture system afterwards for molecular analysis?
So that's exactly what we would like to do with some of the work with mass spec. And so that's really the work that we're trying to do, so where we can identify specific cell populations, see what they look like before they go in the tumoroid model, and then pluck them out afterwards to see how they've changed certain expression and signaling. So that's work that is currently ongoing, and that's really where we want to, that's where we think the model can be powerful, because whereas we can dissect out the cancer mass and the surround, there is still movement of cells between the different compartments. So we want a bit more of a tight, kind of like a watertight argument for how we analyze those different cell populations.
Okay, thank you very much. Thank you for your questions and for taking your time for the presentation and answering the questions. There seems to be no more questions. So Ciro, I hand the floor back to you.
Thank you. So it is now time to present the next speaker. Thanks to Umber, Cheema, and now we introduce Rainer, who is an expert in studying mechanism of intestinal inflammation with emphasis on myeloid cells and epithelial barrier. And today, his talk is on lipid droplet-dependent fatty acid metabolism modulates the polarization of tumor-associated macrophages. Rainer, thanks for coming, and it's your turn now.
Yeah, thank you for the nice introduction, and I apologize for the long title you have to read, but I don't have to read it now, that's good. I share my screen. So as I said, the title I don't have to read again. My name is Rainer Klauben. I work in the gastroenterology department of the Charité in Berlin, and this is our nice building in the south of Berlin. And we started our project about 6 years ago, I would say. And as a lot of people who do something with cancer, I start my talk with this sketch from Hanahan and Whiteberg, which shows the hallmarks of cancer. And as immunologists, we are interested in, when it comes to tumors and to immune cells, is the avoiding immune destruction hallmark. And here we were especially interested in the suppressive myeloid cells and not so much the suppressive T cells. So, and we were asking why or which factors in the tumor microenvironment actually control the polarization of these immune suppressive myeloid cells, which you can find there, to be tumor-associated macrophages or MDSCs. And we were interested in this particular other hallmark of cancer, the deregulated cellular energetics in the tumor. So that means that in the tumor, there are so many different amounts of metabolites and stuff around compared to healthy tissue that we argued this might have an influence on the polarization of macrophages, which are quite sensitive to their environment. So, and if you look at this nice sketch, to the left, you see tumor stages. Here you see fatty acid synthase status of the cells inside the tumor or the tumor cells. And it's pretty clear that if you take the severity of the tumors or the progression of the cancer, the production of fatty acid synthases increases. And that's true for almost all tested tumor entities. And also when you look at a very old paper here from 1966, so this is actually, this knowledge is decades old, you see that in the tumor here, in ascites tumor model, in the tumor you have fatty acids, 4 fatty acids, palmitic, stearic, oleic, and linoleic acid, which are increased tremendously in the tumor model. So we asked ourselves, are these free fatty acids in the tumor microenvironment, do they contribute in any way to the immune suppressive function of these myeloid cells? So we started with a cell line, with the MSC2 cell line. This is a so-called myeloid suppressor cell line, which becomes a suppressive cell line if you add IL-4 to the culture. So what you can see here is a so-called T-cell suppression assay or T-cell proliferation assay. What you see here is a FACS plot of CFSE stained T cells. And with every division of the T cells, the CFSE staining becomes less bright. So every peak here represents one generation of T cells. And it can be clearly seen if you co-culture these T cells with the IL-4 treated macrophages, they proliferate way less than they do if they are not, if the macrophages in the culture are not pre-treated with IL-4. So if we add OLED instead of IL-4, so no IL-4, the first thing we see, they generate lipid droplets inside the cell. But what we can also see, this is the percentage of divided cells. This is the IL-4 positive control. This is just OLED that we get a dose-dependent decrease of T-cell proliferation in these co-cultures just by adding one unsaturated fatty acids. And this is then the control that no endogenous IL-4 actually affects the cells. So how do they do that? Here we could show that as we added an INOS inhibitor, LNMMA, and without this INOS inhibitor, we see a nice inhibition of T-cell proliferation in these assays. If we add the inhibitor, we can rescue this effect nearly completely. So this and many other experiments we performed shows that this is INOS dependent, that is actually about the NO production of these macrophages. So we saw these lipid droplets. What does it mean, or how do lipid droplets get formed inside a cell? We have one key enzyme for that, DGAT or diglyceride acetyltransferase, sorry for that. And what does it do? It catalyzes deacylglycerol into triglycerides and therefore fills these lipid droplets. So if we take these cell lines and add these DGAT inhibitors, we see microscopically less lipid droplets. Also at the FACS, if we stain fatty acids with BodyPy, we see a reduction in the brightness, so reduction in the intensity of the staining, so there's less lipid droplets inside the cell. Here's the statistic for that. But if we co-culture now this inhibitor and OLED treated cells with the T cells, we see again we can rescue this suppressive effect of the OLED treated cells nearly completely. This is then also the statistic for that, so statistic significant effect, and we can also see a reduction in the NO production of the cells. So the DGAT or lipid droplet formation seems to play a role in the suppressive or in the polarization to suppressive cells in this cell line. So these are cell lines, immortal cell lines. They're always quite special. So we thought we look for more primary cells. So we established a model where we generated from murine bone marrow macrophages, and we used the old GM-CSF protocol and added OLED or steroid as a non-unsaturated, so saturated fatty acid control. And what we could see, again, the OLED treated macrophages were able to suppress the proliferation of the T cells in the T cell suppression assay. What you can see nicely at this curve, while steroid has no effect. These are the original CFSE data. And again, it seems to be about NO production. And these cells are, of course, as there are no cell line, they are quite heterogeneous. So when we analyze them further, for example, C11b GA1 staining in the FACS, we found out that if you add OLED, the GA1 positive C11b low population increases. Here's the statistic for that, and we were quite happy about that because until now we actually thought we were working with MDSCs, not so much with macrophages. But when we sorted these two populations and did the T-cell suppression assay with them, we actually saw that the GR1 negative cells are the ones which are suppressive, not the GR1 positive cells in our in vitro system. And also, again, they are the ones who produce high levels of nitric oxide. So we performed an expression analysis for these GR1 negative cells via microarray. First thing we could see, they cluster really nicely here. They have just a small overlap with the steroid-treated group, but mostly the OLE-treated group stands for itself. And we analyzed it, of course, back and forth, but what we found was that we have a clear tumor-associated macrophage signature in these cells. And here's the confirmation for this expression analysis. So the arginase activity is really up, as you can see here with arginase and CD206, a marker for tissue resident, and M2 or non-inflammatory macrophages, however you call them. As you can see here with the MRC1, which is the gene for CD206, is also up. But this is all about metabolism, fatty acid metabolism, what changes actually inside the cell, and not just about some marker regulations or what happens in the cell. Therefore, we performed this mitochondrial respiration analysis. This is the oxygen consumption rate here. These experiments were done with this so-called seahorse analyzer. And OLE has a way, also the OLE-treated macrophages show way higher oxygen consumption rate than the control group. And if we block the DGAT, the enzyme DGAT, and therefore the formation of lipid droplets, we get a reduced oxygen consumption rate in this bone marrow-derived macrophages. We also get, in the audit group, if you add the DGAT inhibitor, you get a reduced amount of CD206 positive cells. And functionally, you get, you can rescue this formation of suppressive macrophages almost completely if you add the inhibitor to the OLE-treated group. So, but this is also true for, or I first asked the question, so what the fatty acids in the lipid droplets are actually used up by the cell. So if we block ATGL and MAGL, two important enzymes to actually get the cell, the lipids, the fatty acids out of the lipid droplets and get them ready to be consumed within the mitochondrium. If you block these, you see also for ATGL a reduction, a slightly reduction in the mitochondrial respiration, and for MAGL a clearer one. But in the CD206 expression, we can see that also in the control group, but all the more in the OLE group, we get a clear reduction of this CD206 positive tumor-associated macrophages. And also functionally, these inhibitor-treated macrophages were not able to suppress the proliferation of T cells. So this was all done in vitro. What about in vivo? So we did a quick and dirty experiment where we injected the DGAT inhibitors directly in an inoculated tumor several times. And we could clearly demonstrate that there is a reduction in tumor growth in this mice. And within the tumor, we could also, so if you add the inhibitor, and within the tumor, you could also see that there is a clear reduction in CD206 positive tumor-associated macrophages. But then again, this is, as I said, quick and dirty because it's not specific. So we could inhibit the TAMs directly, but it could also be that we have here secondary and tertiary effects. So what we did, we asked our Chinese friends from Xihai Xin's lab, from the Institute of Biophysics in Beijing, and they provided us with liposomes in which we encapsulated our inhibitors and which are taken up quite specifically by macrophages. And we treated the mice systemically with these liposomes. We also, of course, have a non-DGAT inhibitor encapsulated liposome as control. And again, we got a statistically significant reduction in tumor growth in these mice, which get the DGAT inhibitor. And we could also show that that's really quite specific also for the tumor side, that we get a reduction in lipid droplet content in the macrophages in the tumor. So do we see actually specifically in the macrophages in the tumors an increase in lipid droplets? And yes, this is just a standard tumor inoculation. If you look in the tumor, you see that the CD206 positive macrophages are the ones with the high lipid fatty acid content, with the high lipid droplet content. And here's also the statistics for that. And you can also find this in the literature. For example, in this nice paper, where they showed that in benign tissue, they do not see ADRP positive macrophages, while in the tumor, the macrophages are ADRP and CD68 as a marker for tumors double positive. ADRP is a structure protein for lipid droplets, and therefore a nice tool to stain lipid droplets in fixed material where you can't stain for the lipids themselves. And we also looked then in tumor samples from patients. We started with the gastric tumor here, and we were quite disappointed because the CD68, CD206 double positive macrophages, they were not ADRP positive. Or what we also want to look, of course, is that there was no difference between benign and tumor tissue, but there were no positive cells at all for ADRP. So we thought, what is this? And just looked for, by chance, for another tumor entity, colorectal cancer, because we're a gastroenterology department. And what we could see that again, for CD68, CD206, there's almost no difference between benign and tumor tissue, but these tumor-associated macrophages, they are just only in the tumor tissue ADRP positive and not in the benign tissue. And we really wondered why is that? Why is there such big a difference? And that's the answer you can see here, data taken from the Human Protein Atlas, that colorectal cancer, also that different cancer entities have different amount of ADRP expression, as shown via histology. And colorectal cancer has one of the highest amounts or the highest amount, and stomach cancer has one of the lowest amounts. So we really took the two opposite poles of these cancer entities for our initial experiments, and could really see that there are obviously less fatty acids around in the gastric cancer than in the colorectal cancer. Therefore, these fatty acids could influence the macrophages less. So as a summary, so OLED treatment alone is able to polarize monocytes and macrophage precursors into suppressive tumor-associated macrophages, phenotypically, but also functionally. You always have to check these effects functionally. This we could show that the lipid droplet derived fatty acid oxidation seems to control this effect. We could show that when we target DGAT as a key enzyme in these TAMs, we can, at least in our models, impede tumor growth. And we could somehow translate this to cancer patients, where we could see a clear difference in the lipid droplet content of the suppressive tumor-associated macrophages compared to the benign tissue, where you could not find these tumors. And this is then also these are the things we're working on right now. We are trying to figure out the DGAT biology by using knockout mice. We want to know what actually happens to the exogenous added oleate, how does it end up in the mitochondrium, which enzymes play a role there. And we are doing a deeper, more comprehensive analysis of the tumors, metabolic, but also, of course, on an immunologic level. And yeah, I want to thank mostly Hervouet. He did almost all the work, but of course the rest of our group and Professor Shin, who made this possible by providing the liposomes, but also helped with some of the mouse experiments. Some of these experiments were performed in his lab, and of course our sponsors. And this is the guy who performed most of the work, and he graduated successfully, as you can see here, and left the lab now, and now new people are working on it. Thank you very much.
So the Q&A is now open, and there's Pankaj who's got a question. So Pankaj, the floor is yours.
Okay. Hello, myself, Dr. Pankaj Kumar Singh. My question is that you have given your most of the work on the oleate. That is, I think, MUFA. Isn't it?
This is what?
MUFA. And whether you have gone through some study on PUFA, PUFA.
Yes. We actually used TOFA once, but we used it in a, this I need to look up. These are really old experiments, but we used TOFA to look into the de novo fatty acid generation, if I remember correctly, and the TOFA did not have a huge impact on our macrophages. That I can remember from these old experiments.
You have not gone through that study. Well, I have already gone through this colon cancer, and I have gone through a very good results in the PUFA. When we use the PUFA, you understand what I am saying?
Yes. But what is the question?
That is saturated fatty acid, I'm saying.
Yes.
Understand? In the colon, it is very much effective. As I have gone through my study, and what your study that is concerned with the MUFA, that is the oleate that you are saying that you have done most of the studies in the oleate, and you have seen the effect of the oleate.
Yes.
And you have gone through the study of the so many organs independently. Is it?
I did not study many of these organs. So this
You have taken the colon the highest.
Yes, that's true.
Okay.
Yes.
Then, one more question I want to ask. You have taken the rat, I think, for your study.
No, it was mouse studies.
Mouse you have used.
Yeah.
Which immunogen you have used to create the cancer in those?
This was a colorectal cancer cell line.
Cell line, okay. You are
yeah, it was a cell line. It was.
Okay.
Because I mean, we wanted to study the immune cells only. We did not care much about the cancer. So this was really as clear a model as possible. Therefore, an inoculation tumor model seemed to be the obvious way. Right now, we are also we want to expand that to genetic models and also chemical inductions. But this was really very basic. We want to see which macrophages we find in a defined inoculation model.
Okay, thank you.
Okay, thank you very much.
Yes.
Ciro, you can go.
Yes. Rainer.
Yes.
So if I got the point, you said that as more metastatic are the tumors, they more have synthesis of fatty acids.
I have to stress these were not my data. This was from the literature.
Okay. And the idea is that these fatty acids are provided to macrophages to induce them to become TAM.
Yes.
Right? And you saw some difference between gastric and colorectal cancer. I'm wondering if it is known whether the cancer-associated adipocytes are also involved in this transfer of, and if gastric and colorectal cancer show different infiltration of cancer adipocytes, cancer-associated adipocytes.
This is a very good question, and but we had not looked into that, and I'm not aware of any literature right now which might be out there, because we did not focus so far really on the cancer cells themselves. This is something we want to look into, that we actually look which cells in the tumor produce the fatty acids, and also of course want to analyze the fatty acid and other fatty acid metabolites content of the tumors. This would be some major proteomics approach, and metabolomics approach we are planning right now, but did not perform that so far.
And you may agree that the cancer-associated adipocytes may contribute to that as well.
Yeah, sure.
Thank you very much.
We're also a gastroenterology group, and we actually, most of our team works on the effect of fatty acids on the fatty adipose tissue, on the inflammation. So we are into that, yeah.
Thank you. And we have Mohammed, who has also a question.
Thank you very much. I have a question is, I mean, these lipid droplets that are responsible for reprogramming the macrophages to TAMs, are they released by the cancer cell to the tumor microenvironment, so all cancer types or all microenvironment associated cells are equally exposed, or are they specifically taken up by the TAM or by the macrophages? If I understood correctly.
Yeah. Now the macrophages form these lipid droplets if there is a surplus on free fatty acids, be it de novo synthesized or exogenous free fatty acids. So our theory is that blood monocytes arrive at the tumor, get in contact with the environment there, which is in some tumors more than in others, obviously, full of free fatty acids, and therefore undergo the procedure we could mimic here in vitro, that they form the lipid droplets and use up this, use up the fatty acids via these lipid droplets and polarize to the more suppressive phenotype, where there are still on this way, of course, a lot of question marks how this happens.
Okay, thank you very much.
Thank you. I don't see any more questions. Thank you, Raina, for your presentation and for answering questions. And Ciro and Danny, you can have the floor.
Okay. Well, this is the last talk. It is really a pleasure to introduce Dr. Ciro Isidoro. Most of us know him personally, and I think it's a great scientist as well as my great friend as well. Dr. Ciro is the professor in University of Piedmont at Novara, Piedmont Orientale at Novara, Italy. And also he's an adjunct professor in so many places, including our university and University of Oklahoma Health Sciences Center, University of Tokyo Dental and Medical School, and Seoul National University, and and so on. And he's a editor and chief of several journals, and also he's a co-editor in several other journals. And he has established, investigated in autophagy and ovarian cancer. Today he's going to talk or share some of his new findings in the autophagy in ovarian cancer context. Ciro, to you.
Thank you, Danny. It's very late, and so I think we won't spend much time to go through the lecture. I have to thank all the speakers and the participants who have been so patient to stay with us. But this is because the lectures were so exciting and really interesting. So I won't spend much of your time now. Just I want to show you what is our main area of work. So we work on how the tumor microenvironment with its soluble factor affect autophagy in cancer cells. And really, I want to scroll down my slides just to give you a message of what we work on. Essentially, we look at how some cytokines, inflammatory cytokines, or like interleukin-6, interleukin-8, or some other factors released by cancer cells or by cancer-associated fibroblasts, how these soluble factors affect autophagy, and by doing that, they influence the behavior of cancer cells, with particular focus on how this autophagy is related with glucose metabolism and chemoresistance and invasion and metastasis. So briefly, autophagy, I guess everybody now knows, is a process for macromolecular degradation. Is done within the lysosome. It starts with the entrapping of the cytosolic material within the autophagosome, and the autophagosome is marked by the presence of LC3. I do not go into details how it happens, but what is important for us is that when autophagy is triggered, the autophagosome forms and entraps within the vacuole the material to be degraded, and this autophagosome is marked by the presence of LC3. The other thing that happens is that the autophagy substrate is marked by P62 sequestosome. So if we follow the fate of LC3 and P62, we can monitor the autophagy pathway, roughly speaking. Essentially, if we see an increase in the LC3 that is bound to the, let me see if I, oh, no, it's here. Yes. So if we see the bound form of LC3, autophagy is on, and if we see degradation of P62, means that autophagy is on. So autophagy in the tumor microenvironment plays a role in keeping the cells in a dormant state, for instance. And you can see here that autophagy can contribute to keep cell dormancy. But, you know, if you have cancer-associated fibroblasts producing cytokines, you can wake up these cancer cells and get relapse. And this is because, and we will show you now, is because the cytokine can downregulate autophagy in cancer cells. So I cannot go. Okay, so the first model I want to show you briefly is how these cytokines can inhibit autophagy. And I will start playing with interleukin-6, which is highly secreted by ovarian cancers. And what we showed is that indeed this interleukin-6 is inhibiting autophagy. And to counteract the activity of interleukin-6, we used this molecule, is a polyphenol, which is shown to be a stronger inducer of autophagy. And indeed, you can see here that interleukin-6 is inhibiting autophagosome formation labeled by LC3 at the migration front, whereas resveratrol is inducing autophagy. And in fact, you see LC3 positive cells at the migration front. And even in the presence of interleukin-6, you can counteract with resveratrol this action. And what happens is that by using this, you inhibit migration. So essentially, interleukin-6 is promoting migration of the cells by downregulating autophagy. And by counteracting this with resveratrol, we can rescue autophagy and we can stop migration. And that it is mediated by autophagy is proven because if we do disrupt autophagy by using spouting, this drug is disrupting Becklin-dependent autophagy, we lose the effect of resveratrol. So indeed, autophagy is opposing cell migration. Interleukin-6, by downregulating autophagy, is promoting cell migration. Resveratrol can rescue this. And here is shown that indeed, resveratrol can inhibit the healing of the wound, but if you disrupt autophagy with spouting, you lose this effect. Then we looked at what was the mediator of this. And so we did some transcriptomic analysis and also some microRNOmic analysis and of the cancer cells treated with interleukin-6 or with resveratrol. And taken together, what we found is that interleukin-6 was inducing a set of microRNAs, and these microRNAs were targeting, we have seen these microRNAs were targeting this protein. This protein is known to interact with Becklin to induce autophagy and keep the cell quiet and dormant. So what happens is that interleukin-6 is inhibiting the production of this protein, and this is the reason why interleukin-6 is inducing cell migration or awakeness of dormant cells, whereas resveratrol is doing exactly the opposite. So we went on to prove that these microRNA indeed do target ARCH1, or DRAS3 is the other name of this protein. And we have shown that, in fact, these protein interact with Becklin-1 to induce autophagy. And so at the end, essentially what happens is that the cytokine, through epigenetic regulation of this protein, is affecting autophagy in cancer cells, and this leads to migration, cell migration. To prove this, we have done also a work on cancer-associated fibroblasts. So what we did was to treat cancer-associated fibroblasts with resveratrol, and what we could find is that resveratrol was inhibiting the secretion of interleukin-6 by cancer-associated fibroblasts. This work was done together with our collaborator in Thailand, where unfortunately this tumor, cholangiocarcinoma, is endemic. And essentially and briefly, I can show you this interesting fact that when the calves isolated from patients with cholangiocarcinoma, these calves stop releasing interleukin-6 and interleukin-8. And so if we use the conditioned medium from calves pre-treated with resveratrol, then we can rescue autophagy, and also we can stop cell migration. And perhaps I can show you here, I don't know if it works, the movie. Apparently it doesn't work, but I'm sorry, but briefly what happens is that if we use the conditioned medium from calves, the cell do migrate, but if we pre-cure calves with resveratrol so that they do not release interleukin-6 and interleukin-8, they do not move. Here is the quantification, and the move doesn't work. That was the more fancy thing. So essentially, this is one part of the work, and then we have proven with other cancer cell lines to show that, again, in the presence of resveratrol, we prevent the EMT, and this by checking N-cadherin to E-cadherin, actually from E-cadherin to N-cadherin transition. And we also showed that even migration through matrigel and transwell, this is mediated by interleukin-6 inhibition of autophagy, which can be rescued if you pre-treat the cancer-associated fibroblasts with resveratrol. I know I'm running, but I just want to give you this strong message that even in vivo with the patient, what we found is that if we measure the presence of interleukin-6 in the stroma, the overall survival of the patient is worse. And this correlates with low autophagy indeed. And then we proved that this is because of chemo resistance, because in this kind of tumor, the drug probably induces autophagic cell death. So you need autophagy at a certain level so that the drug will over-induce stress and precipitate into autophagic cell death. But in the presence of interleukin-6, they become resistant. And then together with Danny, also we looked at the role of glucose in the effect of interleukin-6. And essentially what we found is that interleukin-6 is promoting cell migration in relation to the glucose metabolism and glycolysis. So it is needed for the cancer cells to uptake glucose in the presence of interleukin-6. And this is proven because when we do use interleukin-6 in a medium without glucose, we do not have cell migration. And what does resveratrol, in fact, is to reduce the uptake of glucose, even in the presence of interleukin-6. So another mechanism through which resveratrol can cure cancer-associated fibroblasts and cancer cells is because it is limiting the uptake of glucose, which would be increased by interleukin-6. And in fact, with Danny, we can show that indeed resveratrol reduces glycolysis in these cells, and by reducing this, it can reinduce and restore autophagy, and this is because there is a link between glucose uptake, glycolysis, and mTOR-dependent control of autophagy. And so what happens is indeed that in the absence of glucose, you can rescue autophagy because you remove the brake by mTOR. And so when you have interleukin-6, you have huge amount of glucose and you inhibit autophagy. But when you do not have much income of glucose, and this is what happens with resveratrol, you can rescue. So there are many redundant pathways leading to restoring autophagy when you have resveratrol. On one side, as we said, is through epigenetics, and the other pathway is through control of glycolysis. And this is to show again that indeed glucose is important for interleukin-6 induction of migration and inhibition of autophagy, and I showed you before. And to end up, we did prove that in fact it is because of the mTOR activation or inactivation. You know that mTOR is normally when it is active, it detaches from lysosomes. So actually, when it is bound, when it is inactive, bound to the lysosome inhibits, and when it is activated, detaches and can release the brake. So taken together, we put together the role of glucose to explain and glycolysis. Yesterday we had a nice talk on that because so the glycolysis and link with autophagy, and interleukin-6 is promoting glycolysis, resveratrol is inhibiting. And with this, I wish to conclude. I'm sorry I had to pack everything together, but the take-home message is that we work on this kind of topic. Okay, so with this, I wish to thank, of course, my collaborators, but the big thank goes to all the speakers yesterday, today, and I thank Charlotte for taking care of this webinar. Thanks to everyone.
Thank you so much. Thank you to all.
I've done it in 10 minutes.
Well done.
Okay.
Thank you both to Ciro and Danny for sharing these webinars held over the last 2 days. Thank you to all the speakers, for today's speakers and yesterday's speakers. Thank you to the attendees, and I wish you all happy holidays. Hope to see you all, whether virtual or physical, next year.
Okay. Thanks to everybody. Thank you very much for being with us. Thank you, Charlotte.
Have
Ciao.
a lovely day.
Thank you very much.
Bye.